Hello and welcome to IGCSE Sudarshan Studio. As in the last video we have done with a core paper of IGCSE Biology 0610. Today we are going to solve this IGCSE 0610/42 extended theory paper. Okay, core paper was for the October November series was very easy, and I know as I'm recording this on 23rd Feb 2026, that means tomorrow is a biology theory paper in South Asia. And I wish a very good luck for all the students, those who are writing the Feb March series biology tomorrow, or for all your IGCSE and CBSE or even ICSE examinations. So a good luck for your exam. Let's start our paper here. Okay, so October November 2025 0610. Now theory paper we are going to solve. Instructions once again here. Read all the questions properly. Use only blue ballpoint pen, and wherever the graph is there, please use HB pencil for your diagram and the graph. Okay, next one, write your name, center and candidate number properly in the given boxes. Also answer to each question in a space provided. You can use the rough page also, but please mention your question number there. Do not use any kind of correction fluid or erasable pen. Okay, the erasable pen that we can actually see in the market nowadays. Do not write anything on the barcode. When if you're writing for the board examination, please remember, don't write anything on the barcode. Don't damage the barcode. Fine. Next one, you can use the calculator, but see that you can use the appropriate units while calculating it and giving the final answer. The paper is for 80 marks. The duration is 1 hour 15 minutes, and the total number of marks are represented in the bracket here. The purpose of the QR code, as you can see, this won't be there in the question paper, but it is from my side, so I'll tell you the purpose of the QR code here. Okay, so let's move to the next question. Don't worry, let's move. Okay, the first question, describe what is meant by physical digestion. We have seen in core paper they have asked about the chemical digestion. Some match the following were there. So now in extended they are relating to the same chapter only, but they are asking you the definition or term meaning of physical digestion. Now physical digestion means breakdown of the food into smaller pieces, but in chemical digestion enzymes are involved, chemicals are involved, but in physical digestion there is no such kind. Okay, so it's the breakdown of food, into smaller pieces. Fine. Without chemical change. Okay, this gives you 2 marks. Where is our 2 marks? Without chemical change is your 1M, that is 1 mark, and breakdown of food into smaller pieces is your 1 mark. So this is how your marks distribution is there for 2. Be careful while you are using the words, biological terms, what we expect you in the question paper. Okay, next one. Figure 1.1 is a diagram of a human digestive system, and again state the letter from figure 1.1 that shows one part where physical digestion occurs. Now you know that physical digestion do not change the chemical nature of the food. So where is this physical digestion occurs? One part where physical digestion occurs. The second one, glucagon is produced. Third one, bile is produced. Now bile we will see is it's in the liver. Okay, so the letter is F. Glucagon is basically a pancreas, right? Insulin and all. So this is C alphabet. This is your pancreas. This is a liver. This is the bile duct. Bile is produced. You will write, actually most of you will write E, but remember they are storing it. Bile duct will store, whereas the F, that's a liver which is producing the bile. And physical digestion occurs. Now physical digestion, here you may write A also for option. Fine, but the problem with A is already saliva is mixed with the food and which is called as bolus. Okay, so there is a chemical digestion already there. Physical digestion occurs, may we write here is B. Acids are not yet much involved at present here, so food enters into the stomach and suddenly doesn't react with an acid. It's already been with the saliva, right? So B is the correct answer for the physical digestion here. Okay, moving to the next one. So 3 marks here. Now explain the role of bile in physical digestion. Now bile is very much helpful in fat process, fat digestion, or let's say fatty acids, it will, right? It will digest the fat. So what is that fat digestion is called as? The key term that we use it here is emulsification. 3 marks, 3 points we are looking for. Function or role of bile in physical digestion. So first one is bile helps in digesting fats into fine fatty into small droplets called as emulsification. So our word we got is here, keyword is emulsification. Okay, second one, this process increase the surface area. This process increase the surface area. Okay, why it is increasing the surface area? Increasing the surface area of fat for digestion or for enzyme activity. Okay, for chemical digestion. Okay, third one, this also helps in enzyme activity. So we got enzyme activity, chemical digestion, emulsification. These are our 3 keywords that the examiner is looking for. You can refer your marking scheme whenever you have doubt, so that you have all your keywords and the statement that you can write for the questionnaire. Fine. I hope you got the role of the bile in now physical digestion. One is emulsification, increases the surface area for enzyme activity, and chemical digestion here. Now physical digestion also occurs in mouth, that is breaking down of the food, chewing. State the name and functions of 2 types of human teeth. 2 types of human teeth, they are independently asking any of it. Okay, so in core paper, while we were revising the core paper, I explained you that the dental formula of human teeth is incisor, canine, premolar and molar. So I remember this in this way. I see a PM, Prime Minister, or anything that you can write, that's an acronym for 4 types of teeth: incisor, canine, premolar and molar. Now state the name and functions they are asking. So let's put the first one, incisor. Now incisor is for cutting and tearing the food. The same thing is for canine also. It helps in tearing and You can write slicing or biting the food. So we got the function as well as the name of the teeth, right? So we got 2 marks again here. Let's move. So underlined words are this one, no? These are the keywords that the examiner is looking at. This will help you to get marks. Fine. Okay, next. Figure 1.2 is a photomicrograph of a part of one villus in the intestine, small intestine, and it has been magnified by 800 times. So X is there, Y is there here in the villus. Okay, identify and explain the role of part labeled X and Y in figure 1.2. So they are asking you what is this X and Y. Now in our textbook, for example, the villus diagram or the villi diagram here of the small intestine, if I magnify this, I don't know whether you'll able to see, but you can see these are the small projections here outward. These are hair-like projections. So what are these hair-like projections? We know hair-like projection in small intestine is basically a microvilli. So let's put here, first one is X is identified as microvilli. Okay, and what's a Y here? Y is something which is in the center. Okay, and we know that the center part of the small villi is helpful for absorptions of fats or fat molecules. So that must be a, I'll write here, it's a lactale. You have to imagine the images which is there in the textbook also. The same in human digestion topic, chapter 7, you will find the same structure of a small villi, diagrammatic part, and there you will see a microvilli is there, lacteals are there, and the blood vessels and capillaries which are supplying the blood. So microvilli, so we have identified X part is a microvilli, Y part is lacteal. So let's explain the role, and this should carry 4 marks. That means identification we got 1. Now 2 roles of microvilli we have to explain. What is the role of microvilli? Microvilli increases the surface area for absorption of nutrients. Don't write food here, okay? Write nutrients it is, because food has already been digested there. Fine. So this is one of the example here. So we got increase the surface area 1 point and absorption is the 1 point. So that's how we get 2 marks on this. Fine. Next one. Y is basically a lactale. And what's the function of a lactale? Lactales helps in absorption of fat droplets or absorption of fat droplets. Okay, that's what we have here. So lactales helps in the absorption of fat droplets, whereas microvilli increases the surface area. Okay, you can write absorption, but there's a one more function of lactales, and what is that? They also help in product of fat digestion. Okay, so absorption of product of fat digestion, that is also there. So we'll skip this part and we will just write absorption of fat droplets. Okay, that's what helps in the lactales here. Okay, so moving to the next one here. Again, there's a blank page here. Please remember, use the blank page properly, because on the blank page, if there is some mistake, if you made some mistake somewhere in the question here or while writing the answer, you can strike this off. And how to strike it? It's very simple. If suppose you wrote something here, let's say helps in amino acid uh, production or absorption. Okay, suppose you wrote something here and which you got to know that this may not be the correct answer. So what you're supposed to do, you just strike it. You can just make it in this manner, or you can just strike it like this and mention in the next blank page that this is question number 4, or my bad, whichever is this, by the way, which is this question number, yeah, question number 1, part E. So you can mention here question 1, continuation of question 1, part E, answer. Okay, so you can write continuation of this, and this is how you use the blank page. I hope that is clear, the function or the use of the blank page here. And please remember, this is the QR code which I'm going to explain you in the end of the question paper. And this is what the channel is, IGCSE Sudarshan Studio. Please do support this channel. If you have watched the video half of this, then do like and subscribe, and also share with your friends. Okay, so I think the message is clear now. Moving to the next one, the second part. Dicotyledons and monocotyledons belongs to the plant kingdom and state two main features of plant. The two main features of plants are they are multicellular and they perform. What are they performing here? They perform what? They photosynthesis. Okay, they perform photosynthesis and multicellular organism. Fine. Okay, next one, state the name of two other kingdoms. What are the other two kingdoms? And they are animals and fungi. Okay, I hope you got it here, but if you don't remember, there are other few. That is, we have prokaryotes. So I'll don't write anything else. 2 means 2. Don't put a semicolon or comma and then write. If it is wrong, even actually the examiner will have a very bad impression of you. So don't write anything else. That's it. 2 examples mean 2. But how to remember rest other 3? Remember, the first one is prokaryotes, fungi, plants and animals. This is what we have, 5 kingdom. Protoptist, prokaryotes, fungi, plants and animals. Fine. Double P, F, P, A. Now, figure 2.1 is a diagram of a cross section of a monocotyledon stem. Fine, it's a monocotyledon. We got vascular bundle we have here. Now, describe how the distribution of vascular bundle in a dicotyledon stem would differ from the monocotyledon. So there in the textbook also they have given how the arrangement of xylem and phloem is there, right? And it's only 1 mark. So distribution, so that let's see the key. Describe how the distribution of vascular bundle in dicotyledon stem would differ from monocotyledon. And in dicots, the vascular bundle will be in the ring form. Okay, bundles would be in a ring. Okay, that's what the answer is. Vascular bundle consists of xylem and phloem, so it would be in the ring. That is how the distribution will be there, differs from the monocots. Okay, next one, figure 2.2 is an incomplete diagram and a drawing of a cross section of dicot, sketching the distribution of xylem and phloem, labeling the phloem and xylem and the root hair. Okay, so let's see the root hair here. So this is basically a root hair. Now sketch the distribution of xylem and phloem. Labeling the phloem and xylem is also there. Now how do you distribute the xylem and phloem in this? Any idea? Fine, this is a root hair that we got. Let's see, and it is a dicot root. Okay, so remember this one to give the answer for. Hope you will see first, check out the textbook that can help you out, and remember what was the important thing that we have to do. I hope you got the answer now. Okay. Fine. So let's draw it now. I hope you are still in the recording. Yes. So a pith will be pith in the center, and then with the help of an HB pencil draw the diagram in this way without lifting your pencil. So remember to draw the diagram in a neat and proper way without making much fuss here. Okay, so this is what we have. A center one is the circle. Fine. And after that, what we have here? Done. So the center one is called as pith, and towards the center which is moving here, okay, you can draw like this. Don't shade the diagram, so no shading is allowed. Okay, then this is done, and now this is your pith, so they do not ask you. So PITH, this is a pith center. Towards the pith, no, these basically are xylem. So just you need to no arrows, and this is how you write. Label the image xylem, and this is outward direction is basically a phloem. I hope you got it now. We are not doing any shading work, so such kind of answers, please remember this very important thing. Use only HB pencil, label only with pencil. Okay, next one, do not no shading. No means no. Don't shade the image, it will spoil the previous page also. Okay, so do not do any kind of shading, and the diagram should have only one line in the sense is don't overlap it. Don't no need of overlapping the line again and again. Just a single line, you just need to draw it. That's it. Fine, this is how you can draw the diagram, and that comes with the practice. Try to do this, it's very easy. Try to 3 to 4 times and you will get it. So towards the center is a xylem, and outward is the phloem, and this one is the root hair. So we have sketched the distribution of phloem and xylem, also labeled phloem, xylem and root hair. This will give us a 3 marks. And now the next question is, state two functions of xylem. Xylem, we know that they are always helpful in carrying or transporting water, and what is dissolved in the water is, you guessed it correct, it's minerals or ions. So they help in transport water and transport ions. Okay, next one, A farmer investigated the effect of grinding on the mass of apple fruit produced by apple tree. The farmer removed a 3 mm strip of bark. Okay, so a farmer is investigating the effect of grinding on the mass of apple fruit produced by apple trees. A farmer removed 3 mm strip of bark and phloem tissue from the branches that produce the fruit. So he removed the bark as well as the tissue from the branches. Fine. Grid from the tissue 3 mm here, and phloem completely removed from the base of the tree branch. The farmer griddled 9. The farmer griddled. Okay, this is the griddle word. 9 apple trees and left 9 apple trees ungriddled. So he did not touch the 9 apples, and the 9 apples was griddled here. The mass of 10 fruits from the griddled tree and the mass of 10 fruits from the ungriddled tree were compared. A griddled one was having 1768, ungriddled was giving very less amount of mass. What would be the reason behind this? Okay, let's see. I think Dave should have the question for this one. Describe the result and shown in 2.1 and suggest an explanation for them. Now, for 5 marks, we should see this. What's the farmer is doing here? Farmer is actually taking 9 samples of apple tree, which he has griddled, and other 9 he did not do anything to them. Now, the farmer removed 3 mm strip, 3 mm strip of bark and phloem tissue from the branches that produce the fruit. So here he did where fruits were there, and this is the main trunk which is transporting now, okay, water, and fruits there means photosynthesis taking place at the branches. So griddled one. So just see that, describe the result here. Greater, according to the table, what we can say is griddled tree is producing more mass of fruits comparative to the ungriddled one, right? That means phloem is transported. Okay, fine. So we have to use the data as well as we have to answer for 5 points. So let's see this 5 points, how can we make it. Now first one, to use the data, the griddled part, so we have to mention it 5 points. The griddled plant is producing more fruits than ungriddled plant. Okay, that is how many times? This is mass of 10 fruits, this is 1000, and this is 958. So this is what we have. The griddled plant is producing more fruits than the ungriddled plant. Fine. Compared with the data, I'll use just this one, and 1 second. Okay, compared with the data. Fine. Second point, the griddle part is doing such a thing that, okay, so griddled plant is producing more mass because the leaf performing photosynthesis, okay, the leaf performing photosynthesis, the sucrose which is produced is not transported down, is not transported downward. Fine. So this is what one point is. Now explanation here. See, xylem is transporting the water from the roots. Okay. So xylem is transporting water. That's fine. Water is entering towards this part as well as towards this. So here phloem is being phloem tissue of the branch that produces fruit here. Okay. So water is transported. No impact on the xylem on this side also on the branch. But now remember the end of this one are the leaves which are doing photosynthesis as well as produces starch, and after producing starch the glucose, no, the sucrose, not glucose, sucrose or amino acids. Okay, now the sucrose or the amino acids which are being produced, these are transporting downward, but as the phloem has been damaged, okay, phloem has been damaged here, so the sucrose is not moving towards the main trunk, due to which you will see more number of fruits mass here. So mass of the fruit is more because the phloem has been damaged here and all the sucrose are getting accumulated in the fruit itself. That's why the mass has been increased, and this topic relates with translocation. Fine. Now moving to the third point, leaf are the producers. Fourth point, sucrose is accumulated in fruits. Okay, and fifth one, as translocation we know, sink. Who is sink here? Fruits are acting as sink in which all the sucrose or amino acid is being stored now, and this is how you get 5 marks on this answer. I hope this is clear now. Next one, apples are used to make apple juice, and state one enzyme used in fruit juice production. Apples are very good source of what? Which enzyme? It's pectinase. Biotechnology topic, 21st chapter, the last lesson. It has this. Pectinase are used to digest even the cell wall, right? Pectinase is the enzyme. Next one. So we have done 2 questions here. And now describe what is meant by the term catalyst. Enzyme topic, ATP topic, evergreen favorite topic for the Cambridge year. What do you mean by enzyme here? Now enzymes are biocatalysts, and what do you mean by catalyst? Catalyst is nothing but something that increase the rate of reaction without involving in the chemical reaction, without involving in the chemical reaction. So increase the rate of reaction and they do not involve into it. Okay, so do not change during the reaction also. Next one, a student investigated the effect of temperature on enzyme activity. A starch suspension was kept at 0 degrees Celsius. Amylase solution was added. So a starch suspension is there, enzyme activity, temperature we are checking here, effect of temperature. Amylase solution was added to the starch suspension at 0 degrees Celsius. Okay, sample of the starch and amylase mixture were tested with iodine solution at 1 minute after 15 minutes. So at 1 minute interval we are checking for 15 minutes here. Next one, the student recorded the time at which the starch and amylase mixture stopped turning blue black color. The student repeated this investigation at different temperature, and this is what the result is. At 0 degrees Celsius, the iodine solution stays blue-black. Okay, that means iodine is showing the reaction with the starch. Now at 10 temperature, 10 degrees Celsius, it is 12. Okay, time at which the starch and amylase mixture stop turning blue-black. So 12, 8, 5, 3, 10 and stayed as blue-black here. Okay, so at this temperature it is blue-black, and at 0 degrees Celsius also it is blue-black, but in between it is showing you the time, okay, at which the starch and amylase mixture stop turning blue-black. So this is the time here. So what we have now, explain the result between 0 and 40 in table 3.1. So whenever such kind of explanation is there and it carries 6 marks, always give your answer with support of your data. Use the table to write your answer. Now for 6 marks, 6 to 7 points, let's put it here. So we know that amylase is a enzyme which is going to break down the starch. So we'll just start with this one. Okay, amylase is an enzyme which acts on starch. Okay, in presence of starch, iodine solution turns blue-black. Okay, iodine is brown in color, orange, light orange, and which will turn into blue-black. Now next one, so let's put now the reading here. At 0 degrees Celsius, what is happening? Stayed blue-black. So at 0 degrees Celsius, enzyme break down the starch, so there is same color, that is blue-black. Okay, so basically the amylase is breaking down the starch, and that's why you have the reaction there, blue-black color here. Okay, so one more here we'll do. At 0 degrees Celsius, with this enzyme activity, we can say at 0 degrees Celsius, enzyme and substrate reaction. Okay, enzyme and substrate reaction is low or slow. So we observe the same color. So we got 4 points under this one. Fifth point, now let's put the temperature increases. As temperature increases, what is happening as the temperature increase? The kinetic energy increase. Kinetic energy of particle increases. Okay, increases, and faster breakdown of starch. Okay, so as temperature is increasing, as you can see how less time it is taking for the staying for the blue-black here. Okay, so as temperature increases, even the blue-black, faster the breakdown of starch will happen here. Fine. The next point here, now we'll reach to the last temperature here at 40 degrees Celsius. At 40 degrees Celsius, what is happening? you can see it reaches to its optimum point, and after that it is increased here. So 40 degrees Celsius is optimum temperature further which enzyme denatured. That's it. Fine. This is what we got for our 6 marks question. Okay, so after which the enzymes or the substrates are not able to bind with each other because maybe the enzyme has denatured, and due to which the active sites has been denatured and no further reactions there. Okay, now this investigation was repeated with protease instead of amylase. So protease was there in place of amylase and starch is still there. Explain why the sample from the mixture produce a blue black color at every temperature. We know that this enzyme is not working on starch, right? So what we will mention, protease, mismatch of active site, right? So protease is an enzyme with different active site. Okay, and enzymes are specific to their substrate. So that's the reason there is no reaction. Because of this, there is no enzyme substrate complex or product formation. That's it. That's the answer here. State one factor other than temperature that can affect enzyme action. It is the pH which can impact the enzyme activity. The pH or amount of substrate, amount of enzymes. Fine. These are also other factors which can impact the enzyme activity other than temperature. Moving to the next question, fourth here. State the name of one primary consumer from figure 4.1. So this is an ecosystem food web chart here, and one primary consumer. So a grass is a producer here. One primary consumer is basically very easy. A sage plant is also mentioned here. So you can write these are two producers. A primary consumer you can write as a grasshopper. Even the answer, this is also correct, marmot. This is also a primary consumer. Use the information in figure 4.1 to construct one food chain with 4 tropical level that includes the steeped eagle. So 4 tropical levels we have to write. Okay, we have to include the eagle also into this steepy eagle. So what we will start, always we'll start with the grass. So let's see, first grass, then we have a grasshopper, we have to include even this eagle, right? And then if you move towards this way, marmot can be taken to the 1 2 1 2, then we move to 3, and then the fourth one. Oh god, nice. So we got the first one, grass, then we move to grasshopper, okay, 3, then we move to arrow mark, and then we move to marmot, and from marmot we move to steepy eagle. That's it. 1 2 3 and 4 tropical levels are done, including the steepy eagle here. Now there are fewer gray wolves than marmots in this ecosystem. Explain why the population size of organism decrease from one tropical level to the next tropical level in an ecosystem. Few gray wolves. Okay. Few gray wolves than marmot in the ecosystem. and explain why the population of size of organism decrease from one tropical level to the next tropical level in the ecosystem. So what will be the idea behind this? Population size decreases because at each level, what is happening? At each level, the energy is lost between each no at each level the energy is lost. I think that's enough here. Fine, this is one of the point. The second point would be how the energy, okay, fine. Now suppose if less energy is lost, if less energy is available, then less biomass, correct, can be supported. Now at the example here, energy loss is basically from the grasshopper, grass to grasshopper. Energy is lost in the form of heat, or any other thing is also there, metabolic activity. So this is what we have. Why the population size decreases? Because energy is lost, and according to the 10% law, if you remember, the 10% of energy is only moved to the next tropical level, and rest 90% is lost in the environment for the various metabolic activity, and due to which, if the less energy is available, the less biomass can be supported there. Okay, fine. Moving to the next question here. A figure 4.2 is a photograph of a wild horse, and this horse is a type of a wild horse that nearly became extinct. Fine. After a capitative breeding program, they have been successfully reintroduced into the steppe ecosystem. The capitative breeding program started with 12 wild horses. Explain the risk to a species of a capitative breeding. Means capitative breeding is in the closed environment, in a closed nature. They actually do perform this breeding program very carefully to protect the organism that uses such a small number of individuals here. Now what is the risk behind this? So the risk would be a very less genetic variation. In such breeding, we observe very less variations. Okay, very less genetic variations we are talking about. So this is the first point. Second point, this also increases the chance of inherited disease, because within a few limited numbers only this test is performing, so the chance of transferring the disease also. And the third and very important one is decrease heterozygosity. That is, greater chance that an organism may be a homozygous. So this decreases the heterozygosity, and due to which more number of inherited disease, and we can see very less variation. So such kind of organisms cannot survive for a long time. A hybrid organism is there, but still these are some of the risks of the capitative breeding. There are some other things also, like they may not be suitable for the particular environment. They may be more susceptible towards the various disease transformation or disease transmission also. Okay, and homozygous recessive condition will be there in them. So this is one of the features or risk factors of such kind of breeding program. What are they? They are more susceptible to the transmissible of diseases. Adaptation towards the environment will be very low. That means their immune system will be very low, and that's the reason they can catch the disease very easily. Okay, so 3 points under this. Suggest way of maintaining population of these wild horses in the wild after they're reintroduced into the ecosystem. So how can we protect them, or how can we maintain their number? The first thing is protecting their habitat itself. Protecting their wild habitat. First thing is their habitat itself is to be protected. Second one is we should have laws, that is rules and regulations to protect them from hunting or from poachers. Okay, third and very important one is educate or awareness campaigning. Education and awareness camps to understand their importance in ecosystem. And fourth and very important part one is removal of the invasive, because they are susceptible towards the disease. So the removal of invasive species or invasive which can cause disease to them, because they are more susceptible. So this is what we are. Removal of predators also can be there. Provision of food, proper food or veterinary care. Fine. Monitoring their health, tracking their health. These are also some of the things that we can have to protect these kind of animals. Okay, next is the in vitro fertilization is performed in the capitative breeding program, and this is the image here that we see. An egg is extracted from a female and a sperm is extracted from a male, has been introduced for fertilization, zygote formation, embryo is taking place, and they are now imparted into the uterus lining. So uterus wall, uterus lining. State the name of the process X, Y and Z. X1 is fertilization because zygote is formed. Y is there embryo, so basically this is cell division, and uterus lining, that is implantation. So this is easy here. So first one is fertilization, second one is your cell division, because cell, you can write mitosis also, right, because the cell is dividing, so mitosis, but we'll make it cell division here, and implantation, and that's how we have a 3 marks here. Okay, next one, the fifth one, discuss the advantage of intensive livestock production. The video will be very long, guys. I hope you have patience with this, and I'm solving this from past, I think, 1 hour. Discuss the advantage of intensive livestock production, and I think this is what we have in the end. Oh, we will have one more question to go, and that is complete a fill in the blanks. Okay, let's do it now. So discuss the advantage of intensive livestock production. What is a livestock? Animals. Intensive production means that. Okay, so advantage is this method improves the yield. Okay, next one. First point is this. Second point, less land is required for such production. Fine. Third one, intensive livestock production. That means we are protecting the nature also. That means few of your tree cutting, or you just write few deforestation. Deforestation is less. Okay. Fourth point, or we have already covered 3 points here. Okay. This can also increase the food security. Let's say right here. Okay, discuss, they did not mention how much, so let's say 3 means 3 to 4 we will write. So we'll write here increase the food security. Done. One advantage of intensive livestock production is that antibiotics are often used, and suggest why antibiotics are needed in intensive livestock production. Now antibiotics is to treat the bacterial, right, to treat the bacterial disease. Second one, what will be the second one? Why we are giving antibiotics? Protect them from the diseases. Okay, prevent the treat the bacterial disease, protect them from diseases, that is spreading of bacterial infection. Okay, so one is we are avoiding or the stopping the spreading of the bacterial infection, and second one we are killing the bacteria also. So these are the two things. Okay, so killing and stopping the spread of the infectious disease from the bacteria. C part is figure 1.5.1 shows the change in antibiotics used in the livestock production in 7 countries from the 2010 to 2016. The suggested limit for antibiotic use in livestock is 50 mg per kg of livestock. So the suggested limit is only 50 mg per kg of livestock, and these are the countries that we are using it, like this A to G from 2010 to 2016. So in the year, that means 2010 it was low, 250 mg per kg of livestock, whereas 2016 it was very high with a country. It's very high. Okay, now state the letter of the country in figure 5.1 that used less than the suggested limit. So what is the less? It's 50. So which one has used less than 50? Which country has used less than 50 in 2016? That means the dark one 2016 is D. One is D. Okay, and the second one is F. That is pretty correct. Easy. Calculate the percentage increase in antibiotics used in the country A between 2010 to 2016. So how you will write? So 3 marks is always distributed in this way. So need to show your working. Okay, so 2 significant figure, show your working. So first one is, we'll take A1 is for 2010. We will take first for the year. How much is for 2010? It is 260. 50, 60, and then like this you have for 300. So it is 260, and the unit is mg per kg. Okay, so this is mg per kg. In 2016 year, it is 400. Fine, this is what we have for the first one. Second marks that we get is from the correct calculation. So what we are going to do, we are going to have 140 divided by 260 by 100. What will be the answer here? We get 53.846, and I have to give the answer to the 2 significant figure. That means it should be 54. Calculate the percentage increases, that is 54% increase. Fine, so I hope you got this answer here, 54%. Next one, explain the development of antibiotic resistance in bacteria. How does it increase its resistance? So first thing is bacteria, development of antibiotic resistance. Okay, bacteria are exposed to same antibiotics for a long time. Okay, if they are exposed for a long time, they become resistant. How? Mutations, remember, yeah, resistance due to mutation. Okay, become resistant due to mutation. Antibiotics kills only the non-resistant bacteria. Non-resistant bacteria. That means resistant bacteria will survive, no? So what will happen to this resistant bacteria? The resistant bacteria which has survived will pass the allele to next generation. Okay, so when they are passing this allele to the next generation, that means the new generation will be resistant towards the antibiotics. Okay, that's what we have for the bacterial resistance. So for a prolonged time of the exposure towards the antibiotics, the bacteria becomes resistant either by natural selections or by the mutations, and these bacteria which are now survived due to the exposure from the antibiotics, they will pass their allele and genes to the next generation, and the next generation will increase in their population, and then this is how they become resistant toward the antibiotics. Okay, drugs topic, the best example is what is it? MRSA, MARS, methicillin acetylcholine resistant bacteria. No, MARS. Let me know what is it. Okay, you tell me what is this. MARS, antibiotic resistant, one of the best example which is given in the Cambridge textbook. Okay, so put it in the comment box. What is this MARS? Fine, moving to the next one here. Okay, just give me a minute here. And I made a mistake here. It's not MARS. How can I do this? Okay, fine, happens, man. Sometimes it happens. Okay, MRSA, methicillin resistant Staphylococcus aureus. My bad, it was not MAR Mars kind of thing. It is Mr. MRSA, methicillin resistant Staphylococcus aureus is the super bug, also it is called as. Yeah, this is super bug. Example in the textbook is given, right? Super bug. So my bad, happens sometimes. I'm sorry. Okay, moving to the next one, the last one. I think an athlete monitored their heart rate and breathing rate before, during, and after vigorous exercise. And this is what we have, the graph: breathing rate versus time per minute and period of exercise. As you can see, as the period of exercise increases, okay, what happened? As the period of exercise increases, the graph, that is heartbeat, also increase, breathing rate increases, becomes stationary here at 15 to 20 minutes, and then again it decreases and then becomes normal. Okay, fine. And heart rate during the same time, as you can see, as breathing increases, heartbeat also increase, 15 to 20 stationary, and the same way how it is. Okay, so here, but the breathing rate is 12 and here it is 16. So what is this now? Complete the sentence to describe and explain the result shown in figure 6.1. After exercise, it takes dash minutes for the athlete's breathing rate to return to its resting stage. So what is this? Breathing they are talking about resting. It takes how much to become normal? It takes 20 there to become the normal breathing rate, 22. Okay, after exercise it takes 20 minutes. Then the heart rate decreased by dash beat per minute between 20 to 40. 20 to 40, how much it is? 20 to 40. 20 is 140, that means approximately it should be 150, and this one is 60. So how much you will have now? 0, and this is 14. Okay, and think this much, it's in between, and this is 60, so this should be 40, this is 40, and then no, I don't think this is there, so this is 150 will take, we get 90 here, total it is 90. Next one, lactic acid has built up in dash. Where does it lactic acid builds? We know it builds in muscles. No need of reading the graph there. We know that lactic acid builds in muscle as well as it can build in blood also, causing an oxygen debt. Fine. The heart rate remains high to transport lactic acid to liver. The breathing rate remains high to supplies dash. What it is supplying? It is supplying oxygen for dash respiration, aerobic respiration. AEROBIC, respiration of lactic acid. Now, the carbon dioxide produced during the exercise is excreted, and urea is another substance that is excreted. State the name of the organ producing urea. It's liver. State the name of organ that excretes urea. It's kidney. Okay, please remember, production of urea is in the liver. Excretion of urea is kidney. Okay. Now, balance the chemical equation for anaerobic respiration. Now, we know that in anaerobic respiration, chemical balance means it is utilizing glucose, right? Anaerobic respiration, that means no oxygen involved, which is producing C2H5OH, that is ethanol here, plus of 2CO2. That's it, carbon dioxide. Okay. Now, state one food product that is used that is made using anaerobic respiration in yeast. It's bread. One food product that is made using anaerobic respiration is yeast. And that's end. That's a bread answer here, and end of the question paper. And now the time for the QR. What is the reason behind the QR code? I hope you came to till this page, page number 20, and this is what for the QR is. The purpose of the QR is, and this is a really true thing, that's why I have added up in the end. I know most of the students may not like it, but this is the fact of India, of my village, Maharashtra. The village is in the state of Maharashtra, by the way. So in our village, there is no school. Our small kids have to walk 10 kilometers every day just to get the basic education. My dream or my purpose of starting this channel is to build a school in our village where every child can receive the education they deserve. Will you help? Will you help make this dream a reality? This is not only my dream, but this dream is by the thousand of people, family members, those who are living in the village. Will you help make this dream a reality? Please support our YouTube channel IGCSE Sudarshan Studio educational channel and donate as much as you can. As you are a student, it's up to you, your wish. If you can donate, please scan the QR code which is there on the page number 1 or on the blank page, or even you can share, like, and subscribe to the channel. This will also motivate me to make more videos, content related to IGCSE, because every child deserves a chance to learn and grow. Together we can give them that future that they deserve. So please donate and support from your heart. It's a humble request from my side, from all the villagers from the state of Maharashtra. Jai Maharashtra. Proud to be an Indian, because I'm serving something for my country, doing something for my country, for my village. Thank you. Thank you very much being patient for listening such a long lengthy video. And if you like this channel, please do like, share and subscribe. And if you can support, just scan this QR code and support the channel IGCSE Sudarshan Studio. Till then, see you in the next video. Take a very good care of yourself and your family, because they are the most important people in your world. And a very good luck for your future exams. Take a very good care. Bye.